During the last 40 years, the electron microscope has become one of the most important tools in the study of biology and biochemistry. To understand why, you have to think about the size of cells and molecules. Most cells are several times smaller than the tiniest thing you can see with your eyes. Animal cells, for example, are only about 20 micrometers in diameter. So a millimeter would stretch across 50 of them. To see them at all, you have to use a microscope. Most of the specimens that are studied with a microscope like this, a light microscope, have been fixed and embedded in wax. But using suitable optics, it is possible to look at living cells in a light microscope. These are fibroblasts, connective tissue cells which secrete collagen. They've been magnified several thousand times and filmed with time-lapse photography so that you can see them moving. The most obvious thing inside the cells is the nucleus, with its dark edge. If you look carefully, though, you may also be able to see some thread-like structures in the cytoplasm. These are the mitochondria. They're only a few micrometers long, and it's difficult to make out any details of their structure. You might think it would help if the magnification was increased. All right, have a look at a section of liver. Here it's magnified about 500 times, and it's stained with eosin and hematoxylin to improve the contrast of the image. At this magnification, the image appears crisp, but the individual cells are too small to be seen clearly. You can see the nuclei, they're the dark blobs, but not much else. If the magnification is increased, so that the cells are enlarged to about 1,000 times, the image gets bigger, but you can't make out any more details. This is because the resolution of the microscope is limited to about half the wavelength of the light used. With visible light, this means that the resolution of the light microscope is limited to about 1/5 of a micrometer. So any particle which is smaller than this won't be seen at all. And any two structures which are closer together than 1/5 of a micrometer won't appear as separate structures. The only way to improve the resolution is to use a much shorter wavelength. This can't be done with a beam of light, but it can be done with a beam of electrons, which is what happens in an electron microscope. Moving electrons show wave properties, and the faster they move, the shorter their wavelength. In the electron microscope, electrons are accelerated by a very high voltage, which gives them a wavelength much shorter than that of visible light. In practice, this means that the resolution in the electron microscope is about 100 times better than in a light microscope. Have another look at the section of liver in the light microscope when it's magnified as much as possible. And now look at the section in the electron microscope and see how much clearer the nucleus and mitochondria are. And this is at the same magnification, about 2,000 times. As the magnification is increased, more and more details of the nucleus and mitochondria appear, and you can see structures that you couldn't see at all in the light microscope. The endoplasmic reticulum with its tiny ribosomes. The Golgi body. The lysosomes, they're the dark structures. And the plasma membrane on the left, the outer membrane of the cell, which keeps the cell intact and separates it from its neighbours. Using the improved resolution of the electron microscope, which not only gives clearer pictures at low magnification, but allows the magnification to be increased considerably before the clarity is lost, a whole new world is revealed. Bacteria, which are about the same size as mitochondria, can be seen in detail. Viruses, about 200 times smaller than most cells, can be seen clearly. And it's even possible to see individual molecules. The fine threads on here are molecules of DNA released from a damaged bacterial virus. These photographs, or electron micrographs, were all taken using a transmission electron microscope. In this type of microscope, negatively charged electrons are accelerated by a very high voltage from a heated filament at the top of the column, which acts as a cathode, to a positively charged anode. The stream of electrons emerging from the anode is focused into a beam by a powerful electromagnet called a condenser lens. The electron beam then passes through the specimen and is finally focused and enlarged by a system of electromagnets, the objective, the intermediate and the projector lens, to give an image on a fluorescent screen, like the image on a cathode ray tube of a television screen. The light and dark areas on the screen are related to the dense and less dense areas of the specimen. In an electron-dense area of the specimen, some of the electrons are scattered, so the electron beam arriving at the screen is less intense and the image appears darker. In a less dense area of the specimen, most of the electrons will pass through, so the electron beam arriving at the screen is more intense and the image appears lighter. The electron density of the specimen depends on the atomic number of the atoms that are present. The higher the atomic number, the more the electrons are scattered, and the greater the contrast of the image. Because most of the atoms in biological materials have a very low atomic number, carbon, hydrogen, oxygen, for example, they don't scatter electrons well and don't produce an image with much contrast. To improve the contrast and get an image like this, the specimen is treated with the salt of a heavy metal like uranium or lead. Where the metal stains the specimen, the electron density in that area is increased, producing a much greater contrast in the final image. Using controls at the side of the microscope, the specimen can be moved around in the electron beam so that you can see different areas of it. And if you want to take a photograph of what you see, the fluorescent screen is moved out of the way and the electron beam focused onto a photographic plate or film which is also sensitive to electrons. The whole system, from the heated filament at the top of the column where the electron beam starts to the screen where the final image is seen, is kept under vacuum, so the electron beam isn't interrupted by air molecules. The specimen in its holder must be put into the microscope through an airlock, and it must be specially prepared. The electron microscope can't be used to study living cells because they can't withstand the high vacuum. The specimens must also be extremely thin so that the electron beam can pass through them. This means that either the specimens must be very small, like viruses or protein molecules, or they must be cut into thin sections, 1,000 times thinner than a piece of writing paper. So that they can be put into the microscope, specimens are mounted on copper or nickel grids, which are only 3 millimeters in diameter. Each grid is made up of a meshwork of open squares. The specimen is supported by the metal lattice, while electrons pass through the holes in between. For some specimens, a suspension of viruses for example, the electron density is increased in one of two ways to produce a high contrast image like this. The first, called shadowing, involves coating the specimen with a heavy metal. The specimen is put into a vacuum chamber and then subjected to a beam of heavy metal molecules evaporated from a heated filament. The metal condenses around the specimen, and when viewed in the electron microscope, it casts a shadow, which makes the specimen stand out more clearly. Like these collagen fibrils, from human skin, magnified about 100,000 times. The alternative approach, known as negative staining, involves surrounding the specimen with a solution of a salt of a heavy metal, uranium for example. When the salt dries, it forms an electron-dense layer around the specimen. So the specimen appears light against the dark background. This technique is particularly useful for seeing the surface features on small specimens. These are fragments from the inner membranes of mitochondria. The small knobs sticking out from the surface are formed by the enzyme complexes involved in oxidative phosphorylation. If the specimen is too large to be studied whole, it must be sliced into thin sections. First, though, the structure of the specimen is stabilized by treatment with chemicals, glutaraldehyde and osmium tetroxide, so that as much detail as possible is preserved when the specimen is cut into sections. The specimen is then dehydrated by soaking it in alcohol solutions of increasing strength, and finally it's embedded not in wax but in a block of epoxy resin. It's from a block like this, only a few millimeters across, that the thin sections are cut, using a knife made of glass or diamond. The sections come off onto water and are picked up on one of the special support grids. They can then be stained with heavy metal before being put into the microscope. Usually a number of sections are cut from one block. You can see them coming off the knife like a ribbon. Each section contains a slice of the specimen taken at a slightly different level. So by putting the images obtained from each section together, it's possible to build up a three-dimensional picture which shows the shape of the specimen itself or the shape of structures within it. Because the processes involved in preparing a specimen for electron microscopy are very harsh, there's always a risk that some alteration of structure will take place or that artifacts will be introduced. To minimize this risk, the techniques of freeze fracture and freeze etching have been introduced. In the freeze fracture technique, the specimen is rapidly frozen to the temperature of liquid nitrogen, that's minus 196 degrees Celsius, or even lower. The water in the specimen freezes, which holds everything in place, and the frozen specimen is then put into a vacuum chamber and split with a knife. The specimen always fractures through the line of least resistance, and the fractured face looks like a lunar landscape. If you look at this electron micrograph of the nucleus of a liver cell, you can see the nuclear pores. In the case of freeze etching, the specimen is also frozen, but the temperature is then raised slightly to allow a thin layer of ice to be removed by sublimation. Part of the specimen then stands proud of the surrounding ice. This is shadowed with a layer of heavy metal, and then has a strengthening layer of carbon added to it. The specimen is dissolved away with sodium hydroxide, and the carbon replica is recovered on a copper grid and examined in the microscope. This technique gives quite distinctive results, as in this micrograph of a piece of placenta. Most biological materials can be examined in the electron microscope using one or more of these techniques. Because more structural detail is being revealed all the time, more is being learnt about the form of living things and the relation of form to function, whether in man, microorganisms, or molecules.